Welcome to Applied Photophysics inaugural webinar on the use of objective, statistically verifiable circular dichroism. I'm your host, Keith Solomon, and today's webinar is being given by Dr. Krishna Mallela from the University of Colorado. After a few brief introductory remarks, I'll introduce our esteemed guests and begin the main portion of our program. Before we get started, I'd like to remind all our viewers that our program today will last for approximately 45 minutes, with 5 minutes of introductory remarks, a 30-minute presentation by Dr. Mallela, which will be followed by 10 minutes of questions and answers. If at any point during the presentation you have a query, please submit your question in the provided window. We will be reviewing the questions as they are submitted and will choose the best, most germane questions to pose to Dr. Mallela at the end of his presentation. Also, there will be a very short survey after the webinar is finished, which will pop up automatically for all attendees. So please take a few minutes to complete the survey in order to help us improve this seminar series. First, I'll begin by introducing the sponsor of today's webinar, Applied Photophysics. Applied Photophysics manufactures CD spectrometers that allow for the precise, well-resolved, and reproducible analyses of complex biomolecules. Consequently, the spectra produced by the CharaScan technology platform is quite amenable to mathematical modeling and statistical testing, which in turn allows for objective go and no-go decisions and supports the publication of data with true confidence. For more information concerning the CharaScan technology platform and the Applied Photophysics line of stop-float instruments, please visit us at www.photophysics.com. Now on to the main event. Dr. Krishna Mallela is an associate professor of biophysical chemistry in the Center for Pharmaceutical Biotechnology, Department of Pharmaceutical Sciences, Skaggs School of Pharmacy and Pharmaceutical Sciences, University of Colorado, and Schutz Medical Campus. Dr. Mallela did his PhD in physical chemistry from the Tata Institute of Fundamental Research, India, in 1999. He was a graduate exchange student at the University of California and did his postdoctoral research at the University of Pennsylvania until 2003. Before moving to the University of Colorado in 2007, he was a research faculty at the University of Pennsylvania. Dr. Mallela has more than 50 publications, has been using circular dichroism for studying proteins for 20 years, and his research interests include structure-function disease relationship of dystrophin and utrophin in muscular dystrophy, as well as mechanisms of protein aggregation. So without further ado, Dr. Krishna Mallela will present his seminar, Wonderful World of Protein Circular Dichroism. Dr. Mallela.
Thanks, Keith, for the introduction, and also Applied Photophysics for inviting me to present this inaugural webinar. And this webinar will be followed by several other webinars from both academic scientists as well as the formulation scientists from industry. So we'll be particularly talking about the proteins, circular dichroism of proteins. So before going into real examples, because I just wanted to show 5 case studies where how we used circular dichroism to determine how proteins behave, etc. So we'll just go through some initial introductory slides about circular dichroism. As the name is implicit, circular, that means we are using circularly polarized light. Dichroism, that means 2 colors. Dichroism, colors absorb one over the other. So in circular dichroism, we want to distinguish between molecules which are enantiomers and chiral. So we need to use the excitation source, which is chiral, which is the circularly polarized light compared to linearly polarized light, which is not chiral. So basically what circular dichroism measures is the differential absorbance of the left circularly polarized light and the right circularly polarized light. Circular dichroism is often reported in terms of ellipticity degrees. So if you think about left circularly polarized light here in this diagram as red color and the right circularly polarized light as blue color. So because of the differential absorbance of one or the other, you have a resultant ellipsoid. And theta, the ellipticity angle, is basically the angle between the major axis of this ellipsoid and the line joining the major and minor axis. So expanding the tan theta and then resulting into theta, you can get into radians and degrees. So molar ellipticity is basically, in terms of this angle theta, converted into standard circular dichroism units, degrees centimeters square per decamole. And obviously, which depends, this removes the dependence on the concentration and path length, because here we are using the extinction coefficients. So in the case of proteins, the main chromophore which absorbs in the far UV-CD region, that is between 180 and 260 nanometers, is the peptide carbonyl peptide bond. But the chirality of is coming from the alpha carbon atom, not from the peptide bond. But what we are monitoring is the absorption of the peptide bond. So in this particular far UV-CD region, depending on what is the secondary structure the peptide bond is participating in, we will have significant different characteristics CD spectrum. And the other region where we generally use circular dichroism is the near UV region, where the three aromatic amino acids, phenylalanine, tryptophan, and tyrosine, absorb. And this is where the side chains are not chiral by themselves, but because of the structural environment, because of the rigidity of the structural environment in which these things are located, you can have different circular dichroism spectrum. So since phenylalanine, tyrosine, and tryptophan absorb over different wavelengths, we can particularly focus on particular wavelength regions if we want to determine the tertiary structure around phenylalanine versus tryptophan. So I'll show you some examples. Before going into that, if you see the far UV-CD spectrum of proteins, alpha helix has a particular characteristic negative bands, and positive bands. Particularly alpha helix, you should see negative bands at 208 and 222 nanometers, positive band at 192 nanometers, and similarly beta and other secondary structures. So that means here is the circular dichroism spectra of insulin. So by just looking at the far UV-CD spectrum of the protein, you can clearly see that the CD spectrum has two minima close to 209 nanometers, 222 nanometers, which is very close to expected 208 and 222 for an alpha helix. And then one can easily say that this protein is predominantly alpha helix, alpha helical protein. And the other example, this is the antibody from NIST. And you can see that the circular dichroism has a minimum now at 290 nanometers, very close to that expected for beta sheet. So even without doing any crystallography or NMR, you can clearly say that this protein is a beta sheet protein, in contrary to human insulin. And the tertiary structure, that is the near UV-CD probe. So depending on what is the secondary structure or what is the chirality of the structure around these aromatic amino acids, we can say whether the structure is chiral or not. So I'll show you again some examples as we go on. So these are the examples now, specific examples now I'm going to illustrate from our lab, how we have used various forms of circular dichroism to understand the disease trigger and also the behavior of various pharmaceutical proteins. The first case study I picked is from a disease, the Shane-Becker muscular dystrophy, which is the most common, most severe muscular dystrophy in young children. And there's no cure available for this disease as of now. And so these genetic mutations in a muscle protein dystrophin trigger the Duchenne-Becker muscular dystrophy. And here is the dystrophin colored in blue, green, along red. This is a long snake-like structure. And so we wanted to study what these disease-causing mutations are doing to the protein, whether they're changing the protein structure, stability, or aggregation, etc. And here is the circular dichroism spectra, far UV circular dichroism spectra. and the stability of the mutants, same mutants, when monitored using as a function of temperature as the destabilizing factor. So if you see the circular dichroism spectra, that is the top left-hand panel. So the wild type clearly shows a negative minima at 208 nanometers and 222 nanometers, which immediately says that this wild type protein is an alpha helical protein. But when we introduce the disease-causing mutations in the dystrophin, so that is L54R, A168D, A171P, you can see the disappearance of CD signal at 222 nanometers, and the spectrum is slowly shifting or moving towards 200 nanometers, which is characteristic of a random coil structure, but it's not perfect random coil. And these three mutants are more like a molten globule states where minimal alpha helical structure, but increased random coil structure. Compare the fourth one, that is Y231N, where you still see minima at 208 nanometers and 222 nanometers. So this mutant is still alpha helical, but the other three mutants are significantly not alpha helical, more towards random coil. This is because these three mutants are part of longer alpha helices, whereas this mutant is not part of an alpha helix when we look at the structure of this dystrophin. And so when we measure the protein stability using thermal melts, so wild type unfolds in a cooperative sigmoidal fashion, whereas the other three mutants, L54R, A168D, A171P, you don't see any cooperative melt, again reconfirming the molten globule nature of these mutated proteins. So although Y231N is an alpha helical protein, very similar to wild type, but if you compare the two melts, that is wild type and the Y231N, so you can see that Y231N mutant is significantly destabilized compared to wild type. So our hypothesis is that these disease-causing mutations are perturbing the protein structure, and when they don't, they are basically destabilizing the protein structure by decreasing the stability, thermal stability, or the denaturing stability. Another case study I picked is comparing the two proteins, utrophin versus dystrophin, which are almost 82% similar in sequence. And utrophin has been proposed as a potential drug for Duchenne muscular dystrophy patients because it is ubiquitously expressed even in those patients who completely lack dystrophin. So there are no immunogenicity issues. But the main problem with utrophin drugs is they have much shorter in vivo life. So can we try to find out what is the biophysical reason why utrophin has a much shorter in vivo life compared to dystrophin? So for that, we again use the circular dichroism as well as the fluorescence, and what here we are measuring is the denaturing melts as well as the thermal melts. So here, the top left panel shows the CD, circular dichroism at 222 nanometers, that is the stability of the secondary structure of dystrophin versus utrophin. So this is done as a function of increasing concentrations of urea, that is the denaturant. So you can see that utrophin requires lesser denaturing concentrations compared to dystrophin, which says that utrophin is much less stable compared to the dystrophin. And the right-hand panel, top right-hand panel shows the same data with fluorescence, and the bottom panel shows thermal melts. One can analyze these curves using two-state equilibrium unfolding model to get the Gibbs free energy as well as the M value, that is the linear slope of the linear variation of delta G as a function of urea concentration, which is a measure of accessible surface area difference between the native and unfolded states. Or one can analyze these thermal melts to get the onset temperature or the melting temperature. And we can in addition to the regular spectral changes as well as the stability changes, we can use the staff flow kinetics using for UVCD. And again, these plots are known as the chevron plots. Here what I'm showing is the staff flow kinetics for dystrophin and utrophin, measured by circular dichroism at 222 nanometers and fluorescence. So here red symbols show the data for utrophin, black symbols show the data for dystrophin. As you can see that utrophin is unfolding much faster compared to the dystrophin, which is again consistent with the stability. So because of the decreased stability, increased unfolding rates, we believe that there's an increased proteolysis, and hence the utrophin has much shorter in vivo lifetime compared to the dystrophin. These studies have been followed up with various functional assays, etc., which confirms our hypothesis. But since this talk is mainly on the applications of CD to understand how we use CD to deduce how proteins behave, so I'm only concentrating mainly on the applications of CD. So now I'm going to switch to the near UV CD, where we have used near UV CD as a probe to monitor the changes in the tertiary structure of proteins. This is the case of a protein which has no tyrosines, so only phenylalanines and tryptophans. In the case of folded protein, the blue line here, you can see beautiful vibrational fine structure of phenylalanine as well as tryptophan. And when you compare the native protein with the unfolded protein, you can see that the vibrational fine structure is disappeared in the unfolded protein. And now the near UVCD is almost close to zero. And this protein, when it binds to its substrate, you can see a clear change in the near UVCD spectrum. That is, I'm comparing the blue line versus the red line. And you can see a big change in the phenylalanine region as well as the tryptophan region when this protein binds to the substrate. When we did the same experiment with far UVCD, there were no significant changes in the secondary structure. So good thing is that near UVCD is basically we are monitoring the changes in the protein tertiary structure. So any minor changes, most of the times near UVCD will be able to catch. And the fourth case study I'm going to illustrate is basically here what we are doing is we are using near UVCD to monitor how a chemical modification of a heat shock protein, that is immunoglobulin binding protein, BIP, how it binding to ATP affects because of the chemical modification. So the left panel here is the change in the tertiary structure of the heat shock protein as a function of increasing ATP concentration. The right panel shows the change in the tertiary structure of the chemically modified BIP. So you can quantitate this data and then plot in the case of binding isotherms. And then once you get this data, you write the equations. So that means the observed signal is a sum of signal coming from the free protein and the protein bound to ligand. And then you get the quadratic equation from the binding dissociation constant. So you have the total protein, total ligand, and then you can solve this quadratic equation. You can resubstitute this quadratic root into this equation. And then you can fit the data to get the dissociation constants. So you can see that the chemical modification is basically decreasing this binding of, that is basically KD is going from 2.9 micromolar to 12.9 micromolar. So we have seen that for UVCD and near UVCD, particularly for UVCD, you can use to determine the secondary structure. Near UVCD, you can use to determine the changes in the protein tertiary structure. Then we have seen denation melts, thermal melts, and then we have seen staff flow kinetics. And then we have seen how CD can be used to measure the binding of small molecules to proteins. Now, the last case study where we use near UVCD to monitor the effect of polysorbates on antibody structure. So polysorbates are the primary detergents used in biopharmaceuticals, mainly to prevent the protein aggregation. And so of the 140 marketed proteins and antibodies, most of them have a non-ionic detergent, such as polysorbate 80, 20, or poloxamer. And out of all the antibodies drugs, they used either polysorbate 80 or polysorbate 20. So the question now is what polysorbates do to the protein. So first of all, the mechanism by which these polysorbates prevent protein aggregation is they compete to the air-water interface in the vials with the antibodies or with the proteins. So when there is no polysorbate, both proteins and polysorbates are surface active molecules. So proteins go to the air-water interface, so hydrophobic hydrophilic interface. So the proteins expose their hydrophobic patches, they start aggregating. And so it is one of the main reasons why proteins form aggregates at interfaces. So as we increase the polysorbate concentration, which is a surfactant, which can exist in monomers as well as micelles. So the monomeric polysorbates saturate the air-water interface, so no space is available for the protein molecules on the air-water interface. So there won't be any partial unfolding of the hydrophobic patches. There won't be any protein aggregation occurring because of the interfaces. So these are the two commonly used polysorbates depending on the fatty acid chain, whether it is lauric acid or oleic acid. It's called PS20 and PS80. There's a significant heterogeneity depending on the vendor, depending on the manufacturer. And so you can see that in the head group, particularly polyoxyethylene sorbiton, it can be W plus X plus Y plus Z, can be anything, but the sum should add to the 20. So PS20 and PS80 are used as aggregation suppressors in protein formulations. The typical concentration used in formulations is about their CMC, critical micellar concentration, which is 0.003% to 0.8%. And I listed here three formulations which use high concentration of polysorbates. Particularly Humira, which is the top selling protein drug, uses almost 0.1% PS80. So now the unanswered question about polysorbates is they are excipients, class of excipients. And the definition of excipients is it should be an inactive substance that should not interact with the drug substance. So the question is, do polysorbates interact with proteins? If yes, how do they interact? Because polysorbates are non-ionic detergents. And we know that other non-ionic detergents such as Redonex do interact with pharmaceutical proteins. For a mAb, particularly this study showed that the KD is on the order of 1 micromolar. So we wanted to study, do polysorbates follow similar phenomenon, or whether they interact with the proteins. So we studied this on a Pfizer antibody. So the questions we asked is, do polysorbates bind? How strong is the binding? Where do they bind? Does the binding affect the mAb structure, antibody structure, and stability? Do polysorbates increase the antibody conformational stability? So first we showed that polysorbates protect this Pfizer antibody against aggregation. So this is basically the data from microflow imaging, where we are monitoring the number of aggregates after overnight agitation. So black bars represent antibody aggregates without any polysorbates. So red shows the data with PS80, green shows the data with PS20. And so you can see that the aggregates are significantly reduced when we add polysorbates compared to when there are no polysorbates. Note that the y-axis is a log scale, not a linear scale. So that means here again, PS80 protects better than the PS20. So then the next question is, we wanted to probe whether these polysorbates bind to antibodies. So this is the data from isothermal titration calorimetry. Then when we fit this to the binding equations, you can see that the dissociation constant of the PS20 is about 0.008% weight by volume, with approximately 6 polysorbate molecules binding to 1 molecule of antibody. Whereas in the case of polysorbate 80, the KD is about 0.02% weight by volume, with approximately 3 molecules of polysorbate binding to 1 molecule of antibody. Note that these KD values are in the same range of polysorbates used in formulations, that is 0.003% to 0.8% weight by volume. But the most important thing from KD values and the N values is that polysorbate 20 binds with greater affinity compared to the polysorbate 80, because affinity is the inverse of the KD. Now we wanted to see, we showed that polysorbates bind. And so now we wanted to see how polysorbates, whether they affect the secondary structure or they affect the tertiary structure of mAb. One of the polysorbates, particularly the polysorbate with the double bond in the fatty acid chain, strongly absorbs in the far UV-CD region. So we were unable to do under our experimental setup conditions. We were unable to do the far UV-CD spectrum, so we went on doing Raman scattering. So Raman scattering here, we monitor the secondary changes in the amide 1 band, which is very significant if the protein is an alpha helical or beta sheet or random coil. But you can see that before and after adding polysorbates, we didn't see any change in the amide 1 spectral region in the Raman scattering profile, which shows that polysorbates do not perturb the secondary structure of the antibody. Then we have a beautiful probe that is near UV circular dichroism to monitor the tertiary structure of the antibody. So here are the spectral spectrum near UV-CD spectra of the antibody before and after adding the polysorbate. So black is the antibody with no polysorbates, red is with PS20, green is with PS80. And you can see a very drastic changes in the near UVCD spectrum when we added PS20. And PS80 also shows the change, but it's very minor. Now the question is, when we know that the polysorbates are binding from the isothermal titration calorimetry, no change in the secondary structure, but significant changes in the tertiary structure. The next question we ask is, where do polysorbates bind? Do they bind to the Fab region, that is antigen binding region, or the crystallizable fragment, that is the FC? So we can do this by cleaving the MAB into Fab and FC using papain-based proteolysis. And so we purified the Pfizer basically did the Fab and FC purification for us, and then they gave us the pure Fab and FC samples. So now we know that near UVCD was able to detect the changes in the MAB when polysorbates bind to the MAB. So here is the same near UVCD spectra of the Fab after FC before and after adding polysorbates. So left panel shows the Fab, right panel shows the FC, and you can clearly see that there is a big difference in the near UVCD spectra of the Fab, but we really don't see much change in the near UVCD spectrum of the FC, which immediately says that polysorbates basically are binding to the Fab region, not to the FC region, at least in this antibody, and they perturb the tertiary structure of the Fab, leaving an FC unaffected. And then since we have a probe to monitor the changes in the structure of antibody as a function of polysorbates, we can use the same probe to monitor the stability of the protein before and after adding the polysorbates. Here is the thermal melt using near UVCD as a signal as a function of increasing temperatures. Black is the data with antibody, no polysorbates. Green is with PS80, red is with PS20. As you can see, the new structure that is formed because of the addition of PS20 is unfolding at lower temperatures compared to no change in the global unfolding at the midpoint of this this melting curve. So that means the new structure or part of the MAB is basically less stable or getting unfolded at lower temperatures compared to the unfolding of the complete antibody. And then we can see the same phenomenon when we do the spectral scan near UVCD cyclic dichroism. The left panel is basically the Fab, middle panel Fab with plus PS80, and the right panel Fab plus PS20. So you can see drastic differences, increased differences between PS20 and the Fab compared to differences between Fab and Fab with PS80. And we reconfirmed whatever the observations we made with near UVCD using antibody NMR. And we showed that the near UVCD showed that PS20 causes more structural changes compared to less structural changes in PS80. And you can see the same thing in when we monitor the change in the antibody structure using 2D NMR, and which is also consistent with the isothermal titration calorimetry. So Fab is less stable, but it aggregates less, because the common assumption is whatever the domain which is less stable should aggregate faster or increased. But we see lesser aggregation for Fab because this is again the microflow imaging. And so you can see that the Fab, although it is lesser stable, it aggregates less. And so it is because of the colloidal stability. To confirm this, what we did is we did the Fab aggregation studies in under 2 pH conditions. So pH 5.8 and pH 8. pH 8 is close to the PI. So what we did is in this middle panel, if you concentrate, what we are doing is we are monitoring the change in the for UVCD as a function of temperature. And we are monitoring again the turbidity of the solution using the same chiroscan. You can see that the pH 5.8 solution is unfolding at lower temperatures compared to pH 8 solutions. But if you monitor the turbidity, is the aggregation pattern. So that means the Fab, which is less stable at pH 5.8, is aggregating at higher temperatures. And pH 8, which is more stable, is aggregating faster. The main change in pH is basically we are changing the electrostatic interactions between neighboring Fab molecules. So this experiment is showing that colloidal stability is playing a major role in the MAB aggregation, or particularly Fab aggregation. And then the left panel shows the data from aggregation data from microflow imaging, and the right panel shows just the differential scanning calorimetry, DSC data, showing how the Fab stability varies as a function of pH. So the obvious question is this the only protein where we showed that polysorbates bind to proteins? So there's already one study already published in the literature from Bruce Kerwin's group, where they studied how the polysorbates interact with apoetin, the apoetin alpha. So here is the near UV-CD spectrum of the protein with and before and after adding the polysorbates. So again, you can see the same data is converted to bar chart here. You can see here white bars are PS20, black bars are PS80. You can immediately see that PS20 is causing more structural changes compared to the polysorbate 80. So the conclusion is polysorbates can bind pharmaceutical proteins. Only thing is we need to use the right technique, which is a near UV circular dichroism. So the case studies which we have seen, we have been using protein circular dichroism, and most of our papers have the circular dichroism spectra. So as we have seen, we have to use two probes, far UV-CD to monitor the changes in the protein secondary structure, near UV-CD to monitor the tertiary structure. So simple comparison of the CD spectrum, we can see whether protein is an alpha helical or beta sheet, particularly far UV. Near UV, we can say whether the aromatic side chains are in a rigid structural environment, chiral environment, or whether they're unfolded or exposed to the solvent, freely able to rotate. And we can use the denation dermal melts to determine the protein stability and aggregation. We can use staff flow methods to monitor the protein unfolding, folding, Chauran plots, and the aggregation kinetics and the involved energy barriers. So I only discussed about the electronic circular dichroism, that is far UV circular dichroism corresponding to electronic transitions. But you can use circular dichroism on any absorption bands, such as vibrational, heme CD, which I'm not going to discuss during this talk. So finally, to thank the people who really did the work, the 5 postdocs, Surinder did the major work, followed by Swati, Javier, and the grad students, Resina, Justine, Dinan, and Scott, collaborators, and funding. Thank you.
Thank you, Dr. Mallela, for that wonderful seminar and for inaugurating our seminar series. That was really fine work that you showed us this morning. Now we have some time for questions and answers. Several viewers wrote in some questions that I think are quite pertinent to the talk. So one question is, how do you approach assessing the significance of tertiary structural changes observed between similar proteins by near UV-CD?
So what we use is we use a parameter called weighted spectral difference developed by John Gabriels and Etienne Labs. So what we do is we basically take the difference weight according to the signal at that particular wavelength. That is basically explained in our polysorbates paper. So if the weighted spectral difference is less than 5%, you can say that the structure difference is detected by the near UV-CD are minimal. So compared to the changes which observed, if the WSD or weighted spectral difference is much more than 5%, that's the criterion which we used in the case of polysorbates. Particularly if we use the near UV-CD. So let's go to the slides. So here, basically what we are showing is there are 3 curves for each sample. So black basically has 3 curves. Green has 3 curves there, and then red has 3 curves. So the within the same sample, so the variation, as you can see, is very tight compared to between samples. So we use something called the weighted spectral difference, which was developed by John Carpenter and John Gabrielson.
Thank you very much for that thorough answer to that question. Another viewer writes in, Do you do circular dichroism in the presence of salt? And if so, do you see a lot of noise between 180 and 200 nanometers of UV light?
We do that because basically the buffer absorption below 200, particularly salts, is a major disadvantage, or you won't be able to get a good spectrum for UVCD spectrum. So below 200 nanometers, particularly salts absorb strongly. So, but you can monitor the absorption or the scattering at the same time. And then you can see that when the high voltage HT goes above 1000, so we generally don't consider. But if we want to really get a good spectrum, we try to minimize the salt concentration as much as possible. And we were able to record the circular dichroism spectrum of proteins as low as 170 nanometers using Chiroscan Plus. But playing with the buffer conditions, playing with the protein concentration, playing with the path length of the cuvette may work. You have to really little bit optimize the buffer system and the cuvette and the wavelengths.
Thank you very much. Another viewer writes in, did you verify changes in the secondary structural contents in the presence of PS80 and PS20 to support the Raman data?
As I said, one of the polysorbates absorbed in this for UVCD region, so we didn't do basically for UVCD spectral comparison of this map with and without polysorbates. But maybe if we play with the polysorbate concentration, if we use a shorter path length cuvette, we may be able to record the far UVCD. But what we are more concerned is since the near UVCD gave us the answer that is a change in the tertiary structure. And there is no change in the Raman scattering profile before and after adding polysorbates. So that means there is no significant changes in the secondary structure. But there are very significant changes in the tertiary structure of the map after adding the polysorbates.
Thank you very much. Another viewer writes in a very specific question about one of your experimental conditions. What were the buffer conditions for the ITC measurement for PS20 and PS80 interactions? What was the protein and PS concentrations, so the concentration of the polysorbates?
So if we go to that binding curve, let me pull up the data. So you can see that the molar ratio, and then so we're using obviously the polysorbate, we're starting with the start concentration. And then we are diluting, we are titrating. So we have the protein inside the bulb, our ITC bulb, and then we're using adding incremental levels of polysorbate, and we're monitoring the change in the heat capacity. So you can see that since KD is almost the midpoint of this transition, so we are crossing the midpoint on both levels. So we have a beautiful sort of sigmoidal curve saturating the binding sites and also spanning the KD range. So from the values of this KDs, so you can guess that the ranges which we went is basically 0 molar ratio to almost 14 molar ratio. That is 1 protein to 14 molecules of polysorbate 20 and polysorbate 80.
Thank you very much. Another viewer writes in, when obtaining a delta G unfolding using normalized CD data, do you make the assumption that the unfolding is a two-state process and that there is reversible equilibrium between folded and unfolded states? Very good question.
Yeah, that's the standard question which protein folding field has been asking for a long time. Okay, let's go to that data. So when you don't see a multiple sigmoidal transitions, very few people show like multiple sigmoids, sort of a step function sort of curves. Unless we see that, we don't know whether this protein is a two-state folder or multi-state folder. But the common assumption when we make is we assume that the protein exists in two states, native and unfolded states. But that's just the assumption because you have to start somewhere. So the delta Z unfolding and the M value, which we obtain by fitting these curves, are only qualitative if the protein is a multi-state. And if the protein is a two-state, they are quantitative. So, but if we see a transition, one sigmoidal transition, there is no way of distinguishing whether the protein is a two-state folded or three-state folded. But the M value, since it is proportional to the accessible surface area, if you know the crystal structure of the protein, you should be able to back calculate M value if the denaturant is an urea concentration, urea or guanidine concentration. So you can compare the back calculated M value with the measured M value. And you can, to some extent, you can say whether this protein is a two-state folder or multi-state folder. So when we know clearly that it's not a two-state folder, so the delta Z, whatever we're obtaining by fitting this data to a two-state unfolding equilibrium model, is just a qualitative parameter rather than a quantitative parameter. And also here, in the case of utrophin dystrophin, the melt shows a sigmoidal melt, but you see the unfolding kinetics. So if it is a perfectly two-state protein, it should have only a single unfolding rate. The fact that utrophin shows two unfolding rates and the dystrophin shows two unfolding rates when we measure by circular dichroism, whereas when we measure by, as well as when we measure by fluorescence, clearly this protein is not a two-state folder. And if we look at the crystal structure, it actually has the domain which we are studying here is the actin binding domain, which is made up of two chalcone homology domains. So we already know that it's not a two-state folder, but only we use these denatured melts for a qualitative information. At least even without analyzing, even without assuming the two-state folder, you can see that utrophin requires less denatured concentrations compared to dystrophin, which says that utrophin is less stable compared to dystrophin.
Thank you, Dr. Mallela. We have time for one more question. A viewer writes in, did you try to calculate KD values from the CD data by titrating the substrate? If so, how does it compare to the ITC results?
So the problem is basically near UVCD requires little high concentration. It's almost, if you want to get a near UVCD signal, you had to use almost 50 micromolar protein concentration to get a good signal. So we were unable to do the KD using near UVCD. But if we have enough sample, like the way we have shown in the case of heat shock protein binding to ATP, it's ligand ATP. One can really use the near UVCD data to monitor the binding constant KD. But since we were limited by the antibody samples from Pfizer, we were not able to do the complete binding curve. But at least from this data, you can see that 2 millimolar, that is the saturating concentrations. If you look at the ITC data, you can see drastic changes in the tertiary structure of the Fab, no changes in the tertiary structure of the FC. But I have shown another case where we purified the protein in our lab, that is the heat shock protein BIB. We can use the near UVCD structural changes to determine the KD values.
Thank you, Dr. Mallela. I'm cognizant of the fact that several of you have written in questions for which we do not have time. If you would like those questions answered, we would be happy to submit them to Dr. Mallela after the webinar, and perhaps he could get back to you on his own time to answer your very meaningful, pertinent questions. So thank you, Dr. Mallela, for your time. Thank you all for your time. We really appreciated your attention today. Recordings of today's presentation will be sent to all registrants tomorrow. Please join us again for the next installment of this webinar series, which will focus on the use of CD in biopharmaceutical workflows. In the meantime, if you have any questions about the Chiroscan instrument platform and our other product lines, visit us at www.photophysics.com. And once again, thank you for your attention.